Okay, so welcome to this video. In this video, what we're going to discuss is the endocannabinoids and their role in the cardiovascular system. Okay, so endocannabinoids and the cardiovascular system. So the structure for this video then, what we're going to start off with is a discussion of the endocannabinoid system. Okay, so we're going to look at the main two examples of endocannabinoids, okay, which are anandamide and 2-AG. Okay, then what we'll do is we'll discuss how they're synthesized, how they signal, okay, so the receptors for cannabinoids, and we'll discuss how they're broken down. That's the endocannabinoid system, so basically it's the way that they signal. Okay, and then what we'll discuss is what's the role of the endocannabinoids in the cardiovascular system. Okay, and this is still quite controversial, but we'll gradually build up a picture of what potentially their role is, and as we'll see towards the end, their role seems to be far more important in pathophysiology than physiology. Okay, it doesn't seem as though the endocannabinoid system in the cardiovascular system is usually active. Okay, it's only active really in pathological situations, and it seems to be the case that it's activated to protect you against certain pathological situations. So for instance, it seems to be activated to try and protect tissues against ischemia and also ischemic reperfusion injury as well. Okay, and we'll discuss that towards the end. Right, so let's start off then with a few definitions. So the first definitions that we need to do are cannabinoids, what's a cannabinoid, and what's an endocannabinoid. Okay, so what's a cannabinoid then? A cannabinoid is any molecule, okay, that is capable of having a similar effect to cannabis, okay, and we'll see we'll see much later what the active ingredient of cannabis is, delta-9 tetrahydrocannabinol. We'll have a look at that structure later on, okay, but at the moment that's our definition of a cannabinoid: anything which is capable of inducing a similar effect within the body to the active ingredient of cannabis. Okay, now what then is an endocannabinoid? An endocannabinoid is a molecule that is used within the body. Okay, it's an endogenous molecule within the body that is capable of having a cannabinoid effect. Okay, so having a similar effect to cannabis. So they are effectively molecules that are in our body, okay, and our body can synthesize, which produce a similar effect to this exogenous molecule which is in cannabis, okay, which we'll see later on. Okay, so let's have a look at the main examples of endocannabinoid molecules now. So this list is continuously growing, okay? There is now quite a long list of putative cannabinoids that are endogenous molecules within our body, okay? But there are two main ones that have been studied the most, and it's these two that we're going to focus on in this video, okay? So the two main ones are a molecule that's called anandamide, okay? And the other one is what's known as 2-AG. Okay, so we're going to start off this video then with studying what anandamide actually is and what 2-AG actually is. Okay, so let's start off with anandamide. Okay, so anandamide is one name that is often used for this molecule. However, it's got a more proper name, okay? Its more proper name is to call it N-arachidonyl ethanolamine. Okay, so N-arachidonyl ethanolamine. Okay, and for short, you can abbreviate down N-arachidonyl ethanolamine down to AEA. So A for arachidonyl, E for ethanol, and then A for amine. So you will often, if you read the literature, just see anandamide abbreviated to AEA, and you might be wondering, well, how on earth does anandamide get abbreviated down to AEA? Well, it's because the proper name for anandamide is N-arachidonyl ethanolamine, and it's that name that we abbreviate down to AEA. Okay, so let's try and understand the structure then of N-arachidonyl ethanolamine. Okay, so basically it is arachidonic acid linked onto the amino group, this is the N here, of ethanolamine. Okay, so in order to draw the structure of this molecule out, we firstly need to know what the structure of arachidonic acid is. So that's that we'll look at first, and then we'll see what the structure of anandamide is. So arachidonic acid. Okay, so arachidonic acid is the biologist's name for a molecule that is more properly called all-cis 5,8,11,14 icosahexaenoic acid, and I'll talk you through this name. Okay, so all cis and then 5,8,11,14 icosahexaenoic acid. Now people, if they're going to use this sort of name, often they don't include the all-cis 5,8,11,14 icosahexaenoic acid. What they do is they just call it icosahexaenoic acid. Okay, so you will sometimes just see arachidonic acid called icosahexaenoic acid. More properly, arachidonic acid specifically means all-cis 5,8,11,14 icosahexaenoic acid. So let's think through this name. So firstly, let's start off with this bit over here, icosahexaenoic acid. Well, firstly, let me say that icosanoic acid is just a 20 carbon fully saturated carboxylic acid. Okay, but we don't want icosanoic acid, we want icosahexaenoic acid. Now what that means is that 4 of the bonds in this 20 carbon carboxylic acid are not single bonds, they're double bonds. So this is not going to be a fully saturated carboxylic acid, unlike icosanoic acid. Instead it's going to have 4 double bonds. Okay, now where are those double bonds going to be? Well, this is why it's not a good idea really, unless you know exactly what you're talking about and the other person who you're talking to knows exactly what you're talking about, to call arachidonic acid just icosahexaenoic acid, which is sometimes abbreviated to ETA, because this name doesn't tell you which of those bonds are actually the double bonds. This numbering here tells you which bonds are the double bonds. They're the bonds between carbon 5 and carbon 6, carbon 8 and carbon 9, carbon 11 and carbon 12, and carbon 14 and carbon 15. And this bit here tells you the EZ isomerism of those double bonds. It tells you that they're all in the cis conformation. So you've got 4 double bonds. This is the position of those double bonds. This is the EZ isomerism. Okay, right. So let me now draw this structure out. So we'll draw it as a skeletal structure then. Okay, so we'll start by showing the carboxylic acid group right at the head here. So here's the first carbon, and here is the double bond to the oxygen and the alcohol group coming off it. Okay, now everything else that I draw is going to be in the skeletal structure. I've shown that carbon there simply because, to be honest, carboxylic acid groups look simpler if you draw them with their molecular formula. They look more scary if you draw them with their skeletal formula. Okay, and all of this simplifying things down to skeletal formulae, it's just to try and make the structure look simpler. Okay, so if the skeletal formulae's strict rules don't make it look simpler, then you can do a mixture of a skeletal formula with a molecular formula. Okay, so there's the carboxylic acid group. Then here's the second carbon, the third carbon, so I probably should just remind people of how skeletal formulae work. In skeletal formulae, you don't show carbon atoms, they're implicitly shown as corners, and you don't show hydrogen atoms coming off carbon atoms. Okay, so this here, this is a carbon atom, and it will have 2 hydrogens coming off it, making up the missing 2 bonds here. Okay, so this is carbon 2, carbon 3, carbon 4, carbon 5, and now we know something. Carbon 5 has a double bond coming off it, so between carbon 5 and carbon 6 you're going to have a double bond, and it's going to be in the cis conformation. Now what that means is that on this other side, the important group, okay, which is the rest of the molecule rather than just the hydrogen, is on the same side as this important group. So if you think about these 2 carbons here, if I just draw this out in its molecular formula for a moment. Here are these 2 carbons in this double bond. Now, both of these carbons have 2 groups coming off. They have the important group, which is this portion here of the molecule, and this one has the rest of the molecule coming off here. And then they just have hydrogens. Okay, now, what the cis isomer means is that the 2 important groups are coming off on the same side, basically. That's what cis actually means. It means on the same side. Okay, the other isomer that you could have is the trans isomer, and in the trans isomer, trans means on the opposite side. Okay, so in the trans isomer, the 2 important groups will be on the opposite side. Okay, like so. And then the 2 hydrogens would also be on the opposite side. We want all of these double bonds in the cis isomer form, and therefore they're going to look like this. Okay, so this is the trans isomer form shown below. Right, okay, so all trans 5, 8, 11, 14 icosahexaenoic acid. So let's continue on. So this is the 5th carbon, the 6th carbon, the 7th carbon, the 8th carbon. Okay, now we know that we need a double bond between the 8th and the 9th carbon. Again, it will be in the cis conformation. So 8, 9, 10, 11. Okay, and generally people start to curve the molecule back round like this now, and you'll see why in a moment. Okay, so this is now the 11th carbon, and you're now going to have a double bond between the 11th and the 12th carbon here. Okay, and this is 12th, 13th, 14th, so we need a double bond between the 14th and the 15th carbon, and hopefully you can now see why it's nice to draw it in this way, because you can see that it's starting to become a little bit symmetric here. Okay, so this was the 14th carbon, this was the 15th, 16th, 17th, 18th, 19th, and 20th, so it's almost symmetric here. Okay, and that's why often arachidonic acid is drawn in this way. So this structure that we've just drawn out, this is the structure of arachidonic acid, or to give it its proper name, all cis 5, 8, 11, 14 icosahexaenoic acid, although commonly you will just see arachidonic acid referred to as ETA for icosahexaenoic acid. That will be on the understanding that you realise that we mean that the double bonds are in these positions and they're in the cis isomers. Okay, right. So that's arachidonic acid. So now let's look at the structure of these 2 endocannabinoids. We're on our way to looking at the structure of anandamide. So anandamide is N-arachidonyl ethanolamine. Okay, so this arachidonyl means that that you've got an arachidonic acid molecule within your structure, and we've attached this arachidonic acid molecule onto ethanolamine. Okay, now ethanolamine is merely just an ethanol molecule with an amino group coming off it. Okay, so let me just show you the structure of ethanolamine. So ethanol is just the 2-carbon alcohol. Okay, so I'll do this in skeletal structure again. So here are the 2 carbons. Here is the alcohol group coming off the one of the carbons here. And then to turn it into ethanolamine, you just need an amino group coming off the other carbon. So 2 carbons, one of them has an alcohol group coming off, one has an amino group coming off. So this is the structure of ethanolamine. Okay, right. Now, what we're going to do to create N-arachidonyl ethanolamine is we're going to bind this molecule to the arachidonic acid. Okay, we're going to react this amino group with the carboxylic acid group with an amide link. Okay, and that will create us the N-arachidonyl ethanolamine because we've attached arachidonic acid onto the amino group. That's why it's N-arachidonyl ethanolamine. Okay, so let's draw this full thing out. So we'll start off by drawing the ethanolamine then. So just copying this out now. Okay, so here we go. Like so. Here is the alcohol group coming off this carbon. Here is the amino group with 1 hydrogen coming off it, and now this is going to be attached to the carboxylic acid group here, and I've realised I've just drawn it in an inconvenient way. Okay, so I'm going to redraw it, because if I continue on like this, it's going to ruin the picture, because then I'm going to have to invert the arachidonic acid molecule. I should have drawn the arachidonic acid molecule firstly. Okay, so let me redraw this. Okay, so redrawing the ethanolamine the other way up, simply because it will make the picture look easier. Okay, So I've now redrawn my ethanolamine the other way up. And now what I want to do is attach this onto my arachidonic acid molecule. So here is that carbon of the carboxylic acid group of the arachidonic acid molecule here. Okay, it's now attached to the amino group by the amide link. Okay, so this is how we've attached the ethanolamine to the arachidonic acid, and now I can just copy my arachidonic acid molecule out again. So here's first carbon, second carbon, third carbon, fourth carbon, fifth carbon. We then need a double bond between the fifth and the sixth carbon that's in the cis conformation, seventh carbon, eighth carbon, ninth carbon, again a double bond between the eighth and ninth, 10th, 11th, 12th, double bond between the 11th and the 12th, 13th, 14th, 15th, double bond between the 14th and the 15th, 16th, 17th, 18th, 19th, and 20th there. Okay, right, so this now is the structure of N-arachidonyl ethanolamine, or anandamide. Okay, so this is one of our very heavily studied endocannabinoids. Okay, right, so that's one of them done. Now we'll look at the other one, which is 2-AG. Now we've got a huge head start on this because actually it too contains an arachidonyl group. Okay, so I'm just trying to decide whether I'll do it from the bottom there or go on to the other side. I think I'll go on to the other side. Okay, so 2-AG then. 2-AG stands for, 2-arachidonyl, that's the A. Okay, and then it's now attached onto a glycerol molecule. So 2-arachidonyl and then glycerol. Okay, and that's what the G is for. Okay, so this is just arachidonic acid attached onto the second alcohol group of a glycerol molecule. So let me just remind you of what the structure of a glycerol molecule is. So glycerol is the old biochemist's name, for a molecule that would be more properly called propane 1,2,3 triol. Okay, and although propane 1,2,3 triol is more of a mouthful than glycerol, it's a more useful name because it tells us exactly what glycerol is. It tells us that it's a free carbon molecule with alcohol groups coming off the first, the second, and the third carbons. Okay, so if I draw a skeletal structure of glycerol, glycerol is often drawn with an odd skeletal structure, okay, a very, very straight skeletal structure, which we're going to use. It's often just drawn like this. Okay, a straight line represents the free carbon backbone, and then you just put one alcohol group off there, one alcohol group off here, and one alcohol group off there. Now that's an odd way to draw skeletal structures. Usually, you do it a little bit more spiky, but this is the way that glycerol is usually shown. So this is a carbon, this is a carbon, this is a carbon. Okay, each one of them has an alcohol group coming off. Okay, right. Now what we're going to do is attach an arachidonic acid molecule onto this alcohol group here that comes off the second carbon to create 2-arachidonyl glycerol. And that's going to be done by an ester link. So let's now draw this. Okay, so we just copy the glycerol molecule out again. So here's that third alcohol group, the first alcohol group, and then here's the second one, and now it's going to be attached to the carboxylic acid group of the arachidonic acid molecule via an ester link. So here's the first carbon, here's second, third, fourth, fifth, okay, double bond between the fifth and the sixth, seventh, eighth, double bond between the eighth and the ninth, then the 10th, the 11th, double bond between the 11th and the 12th, okay, 13th, 14th, 15th, double bond between the 14th and the 15th, 16th, 17th, 18th, 19th, and then 20th. Okay, right, so that then now is the structure of this other endocannabinoid molecule, which is 2-arachidonyl glycerol, and it really is just an arachidonyl group attached onto a glycerol molecule. Okay, right. So those are the two main endocannabinoids. As I say, the list of endocannabinoids, putative endocannabinoids, is growing all the time. So there are loads more on top of these that have now been decided to be endocannabinoids. But these two are by far the most studied. So at the moment, at least, they are the most important. Whether they are the most important in physiology and pathophysiology is another thing, but certainly they're the most studied at present. Okay, so we've now seen their structures. Okay, let's now look at their synthesis. Okay, so we're starting to build up this endocannabinoid system, which is all about how are they synthesized, what do they act on, and then how are they destroyed. Okay, so let's discuss their synthesis. So the first thing is, what is the actual trigger that causes the synthesis of endocannabinoids within a cell? Okay, well, it seems to be calcium. Calcium seems to be an extremely important signal that triggers the synthesis of endocannabinoid molecules. Okay, so calcium is the key intracellular signal that triggers the synthesis of endocannabinoids. So if calcium goes up, that seems in some situations within cells to trigger the release, to trigger the synthesis of endocannabinoids, and that's actually an important point, okay? Endocannabinoids, they are not synthesized and stored in cells, okay? They're not like a classical neurotransmitter that you store in synaptic vesicles and then wait to release, okay? No. They are made on demand. Okay, and the reason they have to be made on demand is if we've just looked at the structure of these things, and they are incredibly lipidy molecules. I mean, look at this. This is I should have just drawn your attention to this actually. This is what we might call a monoacyl glycerol. Okay, it is a glycerol molecule with a single acyl group attached to it. Arachidonic acid is just a very special long-chain fatty acid, basically. Okay, and this is just a glycerol molecule with a single long-chain carboxylic acid attached. So it is a monoacyl glycerol. So this definitely is a very lipid molecule. In addition, anandamide to arachidonol N-arachidonol ethanolamine. Again, that's quite a lipidy molecule. Okay, so they're not something that you can store because they can just diffuse straight across membranes. You can't trap them in a vesicle and store them. So they're made on demand. When you want to use the endocannabinoid system, you make them and then release them. Okay, so calcium signals seem to be the thing which drives the production of endocannabinoids, but I want to stress, there are calcium signals going off in cells all the time, and these are not producing endocannabinoids, and it's still not understood how specificity is achieved in intracellular signaling, okay? But there does seem to be some sort of degree of specificity, okay, and there's a lot of interest in microdomains, okay. I'll just sort of say something on this because I think that it makes it difficult to understand why calcium signals don't always trigger the production of everything that calcium signals can when they occur without understanding the concept of microdomains. So microdomains is the leading theory for how specificity is achieved. Okay, so there are all sorts of receptors on the surface of cells, and they all activate the same signaling pathways generally. Okay, so one of the main signaling pathways is the G-alpha Q signaling pathway, which activates the release of calcium from the ER. Okay, now the question is, why, when you get the calcium being released from the ER, don't you get absolutely everything that calcium can do, all being activated at the same time? Okay, well, one of the leading theories is that actually cells are split up into loads of little domains. Okay, so the idea is that effectively a cell is not just one massive great room. In fact, it's split into loads of little domains, basically, by the cytoskeleton making walls, effectively. Okay, and calcium signals in different domains cause different things. Okay, so you can have different effectors for the calcium in different microdomains, basically. So it may be that the synthesis machinery for endocannabinoids is in a specific microdomain, and it's only a calcium signal in that specific microdomain that causes the production of endocannabinoids. Okay, and that's why all the different calcium signals that can act on a cell aren't going to always trigger it to produce endocannabinoids. Okay, it's only when you get signals that trigger calcium signals in a specific microdomain that you actually get the endocannabinoids produced. But that's not proven. That's still very, very controversial. Okay, but it does seem to be that calcium is the leading stimulus for the enzymes which actually produce endocannabinoids. Okay, right. So what are these endocannabinoid synthesizing enzymes? Okay, so once again, we'll start with anandamide and then we'll move on to arachidonyl glycerol. Okay, so what is the enzyme which creates anandamide? Okay, well, it's an enzyme that for short is abbreviated down to NAPE, NAPE, and then PLD. Okay, so what does this stand for? Okay, well, this stands for N-acyl, that's the NA in this name here. Okay, and then phosphatidyl ethanolamine is the PE. So this is phosphatidyl ethanolamine, and once again, I'll talk you through this name in a moment. Ethanolamine. Okay, so that's the PE. So that's overall now the whole of the NAPE portion, N-acyl phosphatidyl ethanolamine. That's that portion. Okay, and then the PLD stands for phospholipase D. Okay, so basically this is an enzyme that is activated by calcium, okay, that is usually attached to the membrane of the cell. Okay, so I'll just draw this here. So if we have our cell here, this enzyme will usually be attached to the underside of the membrane, like so. Okay, and when it's activated by calcium signals, what's going to occur is it's going to start breaking down phospholipids. Well, phospholipids that are in the phospholipid bilayer. Okay, and specifically the phospholipid that it's going to break down is N-acyl phosphatidyl ethanolamine. Okay, so let me discuss the structure of an N-acyl phosphatidyl ethanolamine. Okay, so the basis for this molecule is the phosphatidyl portion. This is the really important portion to understand. Okay, this is the basis for most phospholipids that are in the cell membrane. Okay, so basically this prefix here in front of the ethanolamine implies that the structure contains a molecule known as phosphatidic acid. So I'm going to start off my discussion of the structure of an N-acyl phosphatidyl ethanolamine by discussing the structure of phosphatidic acid. Okay, now we're not going to draw this out with the actual skeletal or molecular formulae. Okay, we're just going to draw a cartoon for this. So phosphatidic acid, I'm going to draw like so in my cartoon. Okay, so let me explain the different portions here. So these two horizontal lines, which I'm now colouring in orange, these represent long-chain carboxylic acids of some sort, okay? So these are long-chain carboxylic acids, and the biochemist name for a long-chain carboxylic acid is to call it a fatty acid. It's just a really long carboxylic acid molecule, okay, that's very hydrophobic. And this is the portion that's going to anchor this into the phospholipid bilayer. So I just draw a picture here. This is the phospholipid bilayer here, and basically what's going to happen is this structure is going to be sitting in the phospholipid bilayer like so. Okay, this just translated over to here. Okay, and these long-chain carboxylic acids are going to be involved in holding it in the phospholipid bilayer. Okay, right, so you've got two long-chain carboxylic acids in this structure there. Okay, the vertical line then that I'm now colouring in green, this is a glycerol molecule, which we've already seen, propane 1,2,3 trial, okay, with those 3 alcohol groups. And indeed, what we have done is attach these two long-chain carboxylic acids onto the first and second alcohol groups of the glycerol molecule. Okay, then onto the alcohol group that comes off the third carbon of the glycerol molecule, we're going to attach a phosphate group. Okay, in purple, and this is why it's called a phospholipid, okay, because the characteristics that you have to have in order to be a phospholipid are you have to have a lipid group, which is made up by these long-chain carboxylic acids here, and then also a phosphate group. Okay, so this is phosphatidic acid. Okay, now this phosphatidyl prefix here tells us that in this molecule we have got a phosphatidic acid. Okay, so what we now want to do is extend phosphatidic acid to build phosphatidyl ethanolamine. Okay, and all we have to do there is attach ethanolamine onto this phosphatidic acid molecule. So what we do basically is we attach the ethanolamine onto this phosphate group of the phosphatidic acid molecule here. Okay, and the way that we do this is we use the alcohol group of the ethanolamine here and attach that onto the phosphate group via what's known as a phosphoester link. So this is the alcohol side of the ethanolamine molecule that is attached to the phosphate group. That's the important bit to understand from that this is the alcohol side and the amino group is free over here. Okay, so that is now phosphatidyl ethanolamine. And then finally, to create N-acyl phosphatidyl ethanolamine, what we're going to do is add on to the amino group of the ethanolamine, which is free over here. We're going to attach on an acyl group, and all that means is we're going to attach on a long-chain carboxylic acid. So another long-chain carboxylic acid is going to get attached on here. Okay, right. And that now is the structure of an N-acyl phosphatidyl ethanolamine shown in cartoon. Okay, now, what then is an N-acyl phosphatidyl ethanolamine phospholipase D enzyme going to do? Well, basically, phospholipase D enzymes always cut phosphatidic acid derivatives like this at this position here. Okay, so they cut this accessory bit that has been added onto the phosphatidic acid molecule off, basically, and they return you the phosphatidic acid molecule. Okay, so one of the products here is clearly the phosphatidic acid molecule restored. Okay, so here it is. So I'll just colour it in again. So here are the long-chain carboxylic acids. Here in green is the glycerol backbone, and here in purple is that phosphate group. Okay, so that's phosphatidic acid returned to us. And now the other thing that we're going to get, and this is the important bit, is we're going to get the ethanolamine here in blue with some long-chain carboxylic acid attached to its amino group. Okay, now I did not tell you what long-chain carboxylic acid that we had attached onto the ethanolamine, and the reality is that it could be loads of different long-chain carboxylic acids. And this N-acyl phosphatidyl ethanolamine, the N-acyl phosphatidyl ethanolamine phospholipase D, it does not distinguish between different N-acyl phosphatidyl ethanolamines. It doesn't care what long-chain carboxylic acid is that you have here. Okay, so it'll cut them all. Okay, so it will produce loads of different molecules here. It will produce ethanolamine attached to all sorts of different long-chain carboxylic acids. Now, in a few of these N-acyl phosphatidyl ethanolamine molecules, it will just happen that this acyl group of the amino group of the phosphatidyl ethanolamine molecule will happen to be arachidonic acid, and it's that we're interested in. So let's say now that this was a very special N-acyl phosphatidyl ethanolamine molecule, and it just so happened that this group was an arachidonyl group. Okay, and then when the N-acyl phosphatidyl ethanolamine phospholipase D cuts here, it's actually going to produce arachidonic acid attached to the amino group of the ethanolamine molecule, and we know what that is. We know that that is anandamide, okay, the N-arachidonyl ethanolamine molecule. Okay, so this is anandamide, or AEA, arachidonyl ethanolamine. Okay, right. So that's how you synthesize anandamide, basically. You activate this N-acyl phosphatidyl ethanolamine phospholipase D enzyme, and it starts breaking down N-acyl phosphatidyl ethanolamine molecules, and some of these, when they're broken down, will just happen to liberate anandamide molecules, and then this anandamide molecule will just be released from the cell, because, as I say, it is lipid soluble, so it can diffuse right through cell membranes. Okay, so you can't contain it once you've produced it. Okay, right, now let's turn our attention now to the synthesis then of 2-arachidonyl glycerol, the other endogenous cannabinoid molecule. Okay, right, so 2-arachidonyl glycerol, you will remember that 2-arachidonyl glycerol was just a monoacyl glycerol. It was just a glycerol molecule with a single long-chain carboxylic acid attached to it that just happened to be arachidonic acid. So if we were going to use this cartoon structure here to show the structure of 2-arachidonyl glycerol, all we would do is we have our glycerol molecule like that, okay, in green here, and then we'd have a long-chain carboxylic acid here in orange, and that long-chain carboxylic acid would just have to happen to be arachidonic acid here, okay, and that molecule would be 2-arachidonyl glycerol. Okay, so how do we produce this then? Well, we produce it actually from diacyl glycerol molecules. So I'll show you a diacyl glycerol molecule here. So a diacyl glycerol molecule is just a glycerol molecule with 2 long-chain carboxylic acids attached to it. Okay, like so. So this molecule here, this is diacyl glycerol, and you have diacyl glycerols present within the phospholipid bilayer. Okay, so again, if we go back to this picture, here would be our diacyl glycerol, and I might just try and add a little bit of colour onto that. Okay, so here are the long-chain carboxylic acids, and here is the glycerol molecule, and then if I have to now reciprocate for the phosphatidic acid molecule. Okay, so here is the long-chain carboxylic acids, and here is the phosphate group. Right, okay. So what's going to happen is you're going to break down diacyl glycerols within the phospholipid bilayer, and this will yield 2-arachidonyl glycerol in the case where you are lucky enough just to happen to have arachidonic acid in this second position here. Okay, so basically this enzyme that does this is going to be a diacyl glycerol, a DAG lipase. Okay, and I should have just mentioned that diacyl glycerol molecules are often abbreviated down to DAG molecules. So the enzyme that cuts one of these long-chain carboxylic acids off here from the diacyl glycerol, and therefore yields just the monoacyl glycerol here with just that long-chain carboxylic acid attached in the second position, that's called a diacyl glycerol lipase. Okay, and again it's an enzyme that is attached to the underside of the lipid bilayer. Okay, right, like in this picture here, and again it's activated by calcium signals to actually perform its function of breaking down diacyl glycerols to yield monoacyl glycerols. Now, if you are lucky enough to just happen to come across a diacyl glycerol molecule that has arachidonic acid in the second position, then the monoacyl glycerol that you will produce here is then the 2-arachidonyl glycerol, this endogenous cannabinoid molecule. Okay, and that's how you can then produce 2-arachidonyl glycerol. Okay, so that's how you can activate then the production of these endocannabinoids within cells. Okay, now what I want to talk about is how they leave cells. Okay, now I've already said that they're lipid soluble, so they can just diffuse right through the cell membrane. Okay, so once you produce them, they can just leave basically. Here is the arachidonyl ethanolamine, anandamide, and here is 2-arachidonyl glycerol. They can just diffuse out of the cell, and they can go and diffuse into other cells. Okay, but also it's worth noting that there is actually a transporter that has been discovered which is capable of actually transporting these across the lipid bilayer and making it easier for them to get across basically. Okay, so they can also end up being transported, and this is a transporter that can transport them in either direction, importantly. Okay, so this is an endocannabinoid, so EC is just an abbreviation for endocannabinoid. Okay, now what is this transporter called? Well, this transporter is called the EMT. Okay, now what does this stand for? This stands for endocannabinoid, that's the E, so I'll just abbreviate endocannabinoid down to EC. Okay, and then the M is for membrane, and then the T is for transporter. So this is the endocannabinoid membrane transporter, which can transport endocannabinoids across the membrane. Okay, right, so that's the synthesis and then the secretion of the endocannabinoids. What we now want to move on to is discussing, now that you've released them into the extracellular fluid, what are they actually going to do? Okay, well, they have receptors basically. the cannabinoid receptors. So let me just get another piece of paper and then I'll discuss these. So let's now discuss the cannabinoid receptors. So there are two main cannabinoid receptors now, and there are a bunch of cannabinoid receptors that haven't yet been fully described as cannabinoid receptors, so it's not fully agreed as to whether they are cannabinoid receptors yet. So there are a bunch of putative cannabinoid receptors as well. Okay, so we're only going to discuss the main two cannabinoid receptors that everyone can agree on. Okay, so cannabinoid receptors then. Okay, so there are two. There is the type 1 cannabinoid receptor, which is abbreviated as CB1, and then there is the type 2 cannabinoid receptor, CB2. So these are read as type 1 or 2 cannabinoid receptors, and that's what the CB stands for, cannabinoid. Okay, right. Now, both of these receptors are G-protein coupled receptors. Okay, so they have the characteristic structure of 7 membrane-spanning alpha helices. So if these two parallel lines represent the inner leaflet and the outer leaflet of the phospholipid bilayer of the cell membrane, then the G-protein coupled receptor structure looks like so. Okay, you have the characteristic 7 membrane-spanning alpha helices, and the amino terminus is always extracellularly and the C terminus is intracellularly. So G-protein coupled receptors, they're a massive great family of receptors. Okay, in humans alone there are over 800 known G-protein coupled receptors. Okay, I think now the total that we found in all life forms goes well over 30,000, and it will continue to grow as we sequence more and more genomes. Okay, so it's a massive great family of proteins, and the features that they all have are that they are a single polypeptide. Okay, so it's one polypeptide, not a great big complex of proteins. Okay, and it has these characteristic 7 membrane-spanning alpha helices. So this is a GPCR, and both the type 1 and type 2 cannabinoid receptors, they are GPCRs, and therefore they look like this. So I'll colour this in purple. Right, so this is a picture of either the type 1 or the type 2 cannabinoid receptors. Now, which G-protein cascade do they actually activate? Well, they activate the GI/0 heterotrimeric G protein. So what does that mean? Okay, well, basically, GI is the most famous heterotrimeric G protein, although there actually are 3 different GI heterotrimeric G proteins. So let me just explain this. Okay, so if I just draw the heterotrimeric G protein here. Okay, heterotrimeric G proteins are so named because they have 3 different subunits, hence trimeric, and the 3 different subunits are different. Okay, so they have an alpha subunit, a beta subunit, and a gamma subunit, like so. So this is our heterotrimeric G protein, which is what's going to be activated by the G protein coupled receptor. Okay, right. Now, there are absolutely loads of different alpha subunits that you can use in heterotrimeric G proteins. There are also loads of different beta subunits that you can use, and loads of different gamma subunits that you can use. Okay, however, it does not seem to make a bit of difference which beta and which gamma subunit you have in the heterotrimeric G protein, i.e. it doesn't seem to affect the signaling functions whatsoever, because beta-gamma subunits seem to do exactly the same thing no matter which one you have in these 2 positions, basically. So it's only the alpha subunit that seems to make any difference as to the actual signaling role of these molecules. Okay, so that's why heterotrimeric G proteins are named according to which alpha subunit they have. Okay, now when I say GI/0, I mean heterotrimeric G proteins where the alpha subunit is either an alpha I subunit or it's an alpha naught subunit. Okay, now there are 3 different alpha I subunits that are all coded for by different genes. There's alpha I1, alpha I2, alpha I3. So those 3 different alpha subunits are all coded for by different genes. Alpha O, there is only 1 gene for alpha O. However, there are 2 splice variants. So there is alpha OA, which is 1 splice variant, and alpha OB, which is another splice variant. Now, basically, you don't need to fuss about this because all 5 of these different alpha subunits, they do exactly the same thing, okay? They inhibit adenylyl cyclase enzymes, okay, and thereby reduce the amount of cyclic AMP. So all of these seem to do the same thing. So that's why we just say that this G protein coupled receptor activates GI/0 heterotrimeric G proteins, okay, because basically that cascade is exactly the same no matter which specific heterotrimeric G protein of these 5 that it activates, basically, okay, and the G protein coupled receptor will actually activate any one of them, basically. It's not specific for, let's say, alpha I1, okay, because all of these pretty much have the same structure in a way, so the G protein coupled receptor will activate all 5 of them. Okay, right, so that's the cannabinoid receptors and their signaling pathways intracellularly. Okay, now, what's then going to happen is that, if we just draw a little picture here, here is cell 1, which might just be producing endocannabinoids. Okay, what can then happen is if we have cell 2 over here, so here's cell 1, here's cell 2, then cell 2 might have a receptor for an endocannabinoid on its surface, okay? So this might be the CB1 receptor or it might be the CB2 receptor. The endocannabinoid can then bind to the receptor, cause signaling cascades within cell 2, okay? And that's how endocannabinoids cause signaling. Okay, now, the final thing then that we need to discuss to complete the endocannabinoid system is to discuss how the endocannabinoids are usually degraded. Okay, so how are they usually broken down? Okay, right, so again, this is different for anandamide and 2-arachidonyl glycerol. Okay, so we'll start off with anandamide. Okay, so basically the enzyme which breaks down anandamide is within the cytoplasm of the cell. Okay, so here it is within the cytoplasm of cells. Okay, and if anandamide just happens to come into the cytoplasm of the cell, so here comes an anandamide molecule, an AEA molecule, this enzyme can break it down. Okay, so what is this enzyme? Well, it's an enzyme that's usually abbreviated down to FAAH, okay, and what this stands for is fatty acid, okay, that's the first FA, and then the AH is for amide, that's the second A, and then hydrolase, okay, so this is the fatty acid amide hydrolase, okay, and this is capable of breaking that bond between the arachidonic acid and the ethanolamine molecule. It's capable of breaking the amide bond. Okay, so if I just get the structure of anandamide back, okay, so here is our structure of anandamide. Here is the amide bond here. Basically, the fatty acid amide hydrolase, it's a hydrolase enzyme. It's capable of bringing in a water molecule, splitting the water molecule up. So if I just show the water molecule here, this is H2O. Okay, it's capable of splitting that water molecule up, binding this alcohol group onto the carboxylic acid group here, binding the hydrogen onto the amino group, and therefore creating our arachidonic acid molecule back again, and then creating the ethanolamine molecule back again. Okay, so basically, fatty acid amide hydrolase catalyzes the conversion of N-arachidonyl ethanolamine into arachidonic acid, which I'll just abbreviate to AA, plus ethanolamine, which I will write out in full. I suppose I could have abbreviated it down to EA, but never mind. Okay, right, so that's the breakdown of anandamide now. Okay. Now let's look at the breakdown of the other endogenous cannabinoid that we've looked at, which is 2-arachidonyl glycerol. Okay, so 2-arachidonyl glycerol is actually going to be broken down by an enzyme which is attached to the phospholipid bilayer. Okay, so I'll draw this here. So it's another enzyme that's attached up here. Okay, and this enzyme is what's known as monoacylglycerol lipase, or for short it's abbreviated as MAGL. Okay, so this stands for monoacylglycerol, MAG, and then lipase. Okay, so this basically is just an enzyme which breaks down monoacyl glycerols. So you can probably guess what it's going to do. Okay, so if we draw our cartoon for 2-arachidonyl glycerol back again, okay, it is a glycerol molecule here in green attached to an arachidonic acid molecule, but it is a monoacyl glycerol molecule. Yes, it's got a very special long-chain carboxylic acid here, arachidonic acid, but it is just a monoacyl glycerol. So basically, monoacyl glycerol lipase will just go, right, you're coming off, okay, and what it will do is it will convert this into an arachidonic acid molecule and a glycerol molecule, so it will break down the monoacyl glycerol. Okay, right, and that's how you can destroy the 2-arachidonyl glycerol molecule, so 2AG here. So that then is how the signal is then terminated. These endocannabinoid signals can be terminated. Okay, so everything that we have just been looking at is what's known as the endocannabinoid system, or for short is abbreviated as the ECS. So at the moment we have not actually seen anything that is specific to the cardiovascular system. Everything that we've been looking at is the same as the way the cannabinoids work in the brain as it is for in the cardiovascular system. We're going to turn our attention to the role of the endocannabinoid system in the cardiovascular system in just a moment. Okay, so this stands for the endocannabinoid, that's the EC, and then the S is for system. So this just refers to how the molecules are made, how they are distributed, what receptors they act on, and then finally how they're degraded. Okay, right. Now, before we actually move on to what these things do in the cardiovascular system, which is the topic for the whole video, okay, what I firstly just want to discuss is the exogenous cannabinoid, or the main exogenous cannabinoid, which is the active ingredient of cannabis. I don't think I could really make a video on cannabinoids without mentioning the active ingredient of cannabis. Okay, so let's just discuss what this does. Oh, and in fact, I probably should have said something that I didn't really stress enough at the time. Okay, I assumed that it was implicitly obvious, but N-arachidonyl ethanolamine, anandamide, and 2-arachidonyl glycerol, they are both agonists at these two types of cannabinoid receptors. So at type 1 and type 2 cannabinoid receptors, they will activate them. Okay, so they're agonists for these receptors. Okay, the active ingredient of cannabis is an exogenous cannabinoid, and it works by also activating these same receptors. So it is an agonist at cannabinoid type 1 receptors and cannabinoid type 2 receptors. Okay, so what is the main cannabinoid in cannabis? And I should probably say that there are actually loads of different cannabinoids in cannabis, loads of different molecules that are capable of activating the cannabinoid receptors. But the main one is what's known as delta-9 tetrahydrocannabinol, or often it's just abbreviated down to THC. Okay, so this stands for tetrahydrocannabinol. Okay, now if you're being slightly more proper, you will call it delta-9 tetrahydrocannabinol. Okay, so you might call it delta-9 THC, or if you're being absolutely strict like a chemist, you will call it negative trans delta-9 tetrahydrocannabinol. Okay, so I want to explain the reasoning behind this name, okay? So I'm going to show you the structure of delta-9 tetrahydrocannabinol, specifically negative trans delta-9 tetrahydrocannabinol. Okay, so basically all of this refers to isomerism. The delta-9 is about structural isomerism, and then the negative trans, I'm afraid, is about optical isomerism grown. Okay, so let's have a look at the structure of negative trans delta-9 tetrahydrocannabinol, which is, strictly speaking, the main active ingredient in cannabis. It's strictly speaking this one that is in cannabis and is the main psychoactive ingredient. Okay, right, so let's draw a structure for it then. Okay, so it has 3 6-membered rings. Okay, so we'll start off with one up here, like so. okay, and there is a double bond in this ring here, okay, and it's this double bond here that is what delta 9 is about. Delta 9 tells you that this double bond is specifically in this position. Okay, in different forms of tetrahydrocannabinol, you can have this double bond in other positions. So that delta 9 tells you that this double bond is strictly there. Okay, right. So that's the structural isomerism portion done. So that's the delta 9. That's what this is referring to here, this double bond. Okay, then you've got a methyl group coming off up there. Okay. Then you've got another 6-membered ring coming off down here with an oxygen right at the bottom there. And then you've got a benzene ring sticking off the side here, like so. So alternating double and single bonds in the Kekulé model. Okay. And then we've got an alcohol group coming off here, and then a 5-membered carbon side chain off here. Okay, like so. And then finally you've got 2 methyl groups coming off here and here. Okay, now so far we have discussed the delta 9 portion. Okay, the delta 9 portion tells you that that double bond there is there. Okay, in other forms of tetrahydrocannabinol it can be at other positions. Okay, so that's delta 9 tetrahydrocannabinol. Now what I want to explain to you is the negative trans delta 9 tetrahydrocannabinol. So, this molecule that I have drawn for you, which is delta 9 tetrahydrocannabinol, has multiple optical isomers. Okay, now let's think about this. Where are the chiral centers? Okay, well, the chiral centers, there are 2 chiral centers in this molecule. Okay, remember, chiral centers need to have 4 different groups coming off them. Okay, now there are only 2 chiral centers in this molecule, and they are here and here. Okay, these 2 carbons here have 4 different groups coming off them. Okay, if we look at this one, for instance, this is one group, this is another group, this is another group, and then the final one is a hydrogen. Those are 4 different groups, and it's the same story for this one here. Okay, so that means that there are 2 possible conformations for this, 2 different arrangements of the groups around each of these carbon atoms. Okay, now let's try and understand this. So if we just draw out that little junction again, okay, so here is the top of this portion here. I'm drawing it for this carbon here, so this is, I might actually use a different colour than blue. This carbon here is this carbon here, and those 2 bits are those 2 bits. Okay, let's imagine that these 2 groups are in the plane of the piece of paper. So this carbon down here is also sitting in the plane of the piece of paper, and this carbon here is sitting in the plane of the piece of paper, and this green one is sitting in the plane of the piece of paper. So all of that's in the plane of the piece of paper. And then the final 2 groups that come off that turquoise carbon, one of them has got to go into the page away from us, and the other has got to come out of the page towards us. Now the question is, which is it going to be? Is it going to be this major group here with all of this hanging off it that's going to be coming out of the page, and then the hydrogen that we haven't even shown on this diagram will be going into the page away from us, or are we going to have it the other way around? Okay, well basically, this negative portion here tells you that the hydrogen is the portion that's going into the page away from us, which I'll abbreviate like so, and then this carbon that's part of this ring up here will be coming out of the page towards us, and the way that I show bonds coming out of the page towards us is by it getting thicker like so. So the carbon comes out of the page towards us, the hydrogen goes into the page away from us. That is what this negative refers to. If you had it the other way round, with the hydrogen coming out of the page towards us and the carbon going into the page away from us, that would be a positive. You put a positive there. Okay, right. Now the positive forms of delta 9 tetrahydrocannabinol do not exist in the the natural cannabis plant. Okay, so you can make them chemically. Chemists do make them, but they're not naturally occurring. Okay, right. Now, then we turn our attention to the other one, and it's the exact same situation. This again has 2 arrangements. So again, we can imagine that the carbons here, okay, and again I'll colour code it so that it's easy to understand. Let's colour code this one purple. That's that carbon there. Imagine that all of these 3 carbons, which are the carbons of this ring here are in the plane of the piece of paper, again we have the same crisis. We have to decide, is this carbon going to come out of the page towards us and the hydrogen going to go into the page away from us, or are we going to have it the other way around? Okay, well basically, unfortunately, the naming isn't quite as nice as for this one. Okay, it gets more complicated because now we go over to naming it with cis or trans. Now what does this mean? Well, basically, the fact that this is in the trans conformation means that the hydrogen that comes off this one is going to be in the opposite direction to the direction that the hydrogen comes off this one. Okay, so that means that the naming of the orientation of this one depends on how this one was oriented, which in my opinion is just stupid. Okay, it makes it more complicated than it needs to be, but that's the way we do it. So basically, negative trans, the negative tells us that the hydrogen off this one here will be going into the page away from us. The trans then says that the hydrogen off this one will be doing the exact opposite to the hydrogen off this one. So the hydrogen off this one therefore has to be coming out of the page towards us, going in the opposite direction to the hydrogen off the turquoise one. And that means that this carbon then ends up going into the page away from us. Okay, like so. So if I wanted to actually draw this on this structure, what I would do is I'd show the hydrogen here going into the page away from us, and the hydrogen here coming out of the page towards us. And then you would imply then where these other carbons have to go from where the hydrogens are going. Okay, so that is the structure of negative trans delta 9 tetrahydrocannabinol, basically. Okay, and that's the specific optical isomer of delta 9 tetrahydrocannabinol that is present within cannabis. Okay, so basically this molecule, negative trans delta 9 tetrahydrocannabinol, is an agonist at both type 1 and type 2 cannabinoid receptors, and therefore does the same thing as the endogenous cannabinoids. Okay, it would be referred to as an example of an exogenous cannabinoid, okay, because it's exogenously applied. Okay, it's not something that you would find endogenously in the body. It's from an exogenous substance such as cannabis. Okay, right, now the one final thing that I want to say here, which is just on the subject of the general endocannabinoid system, before we then go specifically onto the role of this in the cardiovascular system, is that anandamide also has another important target. Okay, and this is going to be really important in the effects of this on the cardiovascular system. Okay, it has another target, and this other target is TRPV1. Okay, so let me tell you about this. So TRPV1 is completely different from what we have seen previously. Okay, the cannabinoid receptors CB1 and CB2, they were G protein coupled receptors. TRPV1 is now a non-selective cation channel, and is famous for its role in pain sensation. Okay, specifically heat pain sensation. Okay, so what does it stand for firstly? Okay, it stands for transient, that's the T, receptor, that's the R, and then potential, transient receptor potential, and then it's vanilloid because it can be activated by molecules known as vanilloids. Okay, vanilloid 1. So it's the transient receptor potential vanilloid 1 receptor, or the transient receptor potential vanilloid 1 channel. People would generally call it the TRPV1 channel. Now, what is it then? It sits in cell membranes. So if I draw a cell membrane here, this is the TRPV1 channel, just drawn very crudely. And basically, when it opens, so this is it closed, when it opens, I'll now draw it open, it forms a non-selective cation channel, which means that it allows sodium ions, potassium ions, and calcium ions to move through it. Now, what are the key movements that are then going to actually occur through it? Well, the key movements that are going to occur is that you're going to bring sodium ions into the cell, and also that you're going to bring calcium ions into the cell. Okay, so this is generally going to cause depolarization when you open the TRPV1 channel. Okay, it also brings in calcium ions, and that's going to be important later on. Okay, so this is what we would describe as a non-selective, oops, is it going to fit in? Just non-selective cation channel, which can often be abbreviated down to NSC channel. okay, non-selective cation channel. Okay, right, so I'll just colour it in and then I'll explain what anandamide does to the TRPV1 channel. So basically, anandamide, but not to arachidonol glycerol and not delta-9 tetrahydrocannabinol, okay, activates the TRPV1 channel, okay, so that's why TRPV1 is not considered a cannabinoid receptor, because it's not a receptor for the actual active component of cannabis, and after all, that was the whole starting point for the definition of the cannabinoids, that they mimic cannabis's effect. Okay, but anandamide, not only is it an agonist at the cannabinoid receptors CB1 and CB2, but it also actually can bind to TRPV1 receptors and cause them to open. So anandamide is an agent which is capable of activating TRPV1 receptors, okay, but not the other ones, so not to arachidonol glycerol, the other endocannabinoid, and certainly not delta-9 tetrahydrocannabinol. Okay, right, and that will become important later on when we discuss the role of anandamide in the cardiovascular system. Okay, so we'll call it there for this video, and in the next video what we will do is we'll start actually looking at how this plays a role in the pathophysiology of the cardiovascular system.